finish the multiple alleles lesson focusing on blood types. Let's take a look at this next portion here. So take a look at this figure, and I want you to try to think about this question. So in addition to OA, B, AB blood types do have a second designation. What is a second designation, and what seems to cause it? So analyze these blood types here, and you'll notice that there's something else that you can use to classify blood types. So if you're analyzing it carefully, looking at the differences, you'll see there's O minus, O plus, A minus, A plus, B minus, B plus, A, B minus, AB plus. And what you'll see is that what they have in common is the carbohydrates on their red blood cells, so A, B, AB. But what's different is that the A plus has this, or the pluses have this RH factor in addition to the carbohydrate, if they have one, on the red blood cell. And so that RH factor, if you have it, that makes you positive. And if you don't have it, you're negative. So there's negative, you don't have that RH, and positive, you do have that RH. And that's called the rhesus factor, and we'll see more information about that in just a minute. Now that part there, RH, is actually genetically determined. You can be RH positive or RH negative. Typically, we'll represent RH by the letter D. So let's take a look at this. This is called a pedigree over here. This is basically just showing a mating between two individuals, and they produce some children. These two individuals here, this person is homozygous for the RH plus, meaning they have two RH plus alleles. And this person is homozygous for the RH minus. They have two RH minus alleles, as you can see here. And when they have kids, you'll notice all the kids are plus. So the plus and minus individual that were pure breeding made kids that were only plus. So from there, what can you conclude about the dominance? Is RH plus dominant to RH minus, or is it the other way around? So hopefully you're able to figure out that since this individual has the RH plus and RH minus allele, and they're plus, in the end, this means that this must be dominant. So RH plus actually is dominant. That is called the rhesus factor. It's a protein on the red blood cell. And basically, if you have the protein, you're positive, you're RH plus. If you don't have it, you're negative, RH minus. So if you're A positive, it means you have the RH factor. But if you're A negative, it means you do not have the RH factor. And as we mentioned, this does follow simple Mendelian genetics. The RH plus is dominant to the RH minus. So it's not quite a multiple allele situation. This is simply showing you a character, which is the RH plus or the RH factor. And it is governed by simple dominance, complete dominance in this situation. And the reason we're just covering it here is because it relates to your blood types as well. So you can do Punnett squares with this, and generally speaking, for the RH plus, we'll represent it by the letter D uppercase, and for the RH minus by the letter D lowercase. So if you look here, you can see we have an RH plus homozygous father, RH minus homozygous mother, and they make 100% RH plus children. And then over here we have a cross between a heterozygous RH plus father and a homozygous RH minus mother, and you can see some of the children will be RH minus, and some will be RH plus. And so over here you can see that we have the carbohydrates on the red blood cells. So we have the A carbohydrate, and then we have the B carbohydrate on the B, and we have the A and B on the AB. And then you can see for the minuses, you don't have the rhesus factor, but on the pluses, you do have the rhesus factor, like that. So let's go take a look at why we kind of need to care about that RH plus and RH minus. So what can sometimes happen during pregnancy is something called RH disease. So suppose that there's a mother who's expecting a baby, and that mother is RH negative. Suppose that their baby, due to genetics, as you can see here, right? Here's an RH negative mother, and here are some RH positive children they can have. So they have an RH positive baby there. What's going to happen is that RH positive baby is going to produce the Rh positive protein, the rhesus factor. And that will actually enter the mother's bloodstream. Now, when the mother has something enter her bloodstream that she's not used to, for example, Rh protein, she wouldn't be used to that because she's Rh minus, minus all around here. What's going to happen is her immune system is going to start to create special cells to basically get rid of that Rh protein. So those special cells, those special components, I should say, parts that are being produced by the mother are called Rh antibodies. And basically the Rh antibodies are going to destroy the Rh positive proteins. So that sounds like bad news, right? So for the first baby, it's okay. For the first baby that's Rh positive in there, it's going to cause the mom to produce these Rh antibodies. Now, if the mother is pregnant again with another baby that's Rh positive, these Rh antibodies are basically going to attack the Rh factor proteins on the baby, and it could really severely injure it. So basically, to prevent that, what we do is we give a RhoGAM injection, which is a way to prevent the mother from forming Rh antibodies in the event that she has a baby that's Rh positive and she's Rh negative. So that's pretty good at preventing that issue from happening, and doctors are pretty good at checking for that if there's any issues with that. That's called Rh disease, and it just highlights how we have these genetic factors that interact with our body systems, such as our immune system in this situation here. So it's important to study genetics because all of our instructions are coded for by these genes, and they determine the interactions within our body and what diseases we have and what our outcomes are. So it's quite important to know that stuff. So let's take a look over here at a sample problem that involves Rh factors. So we have two oparents that have a child. One is homozygous Rh plus, while the mother is Rh minus. Determine the ABO blood type of the child. So first, we're going to start with just try to find out if the child is A, B, O, or AB. Now, the two parents are O, so you should be able to tell that by now that that's a cross between II. And so if you do your Punnett square, you get II, II, II. All they can have is type O. So that child is going to be type O for sure. Now, we want to determine the Rh factor. Okay, so if I ask you a question where I want you to tell me the blood type and the Rh factor, deal with one character at a time. Focus with the blood type first, and then do the Rh factor after. So we told you that the father is Rh positive homozygous, so big D, and the mother is Rh negative. We didn't say homozygous or heterozygous, but since you know the Rh negative is recessive, you know that that's little d. So in this situation here, if we do our Punnett square, you'll see that the only child they could make is big D, which means that they have to be positive. They're heterozygous, but they're still positive. So in the end, the baby is going to be O positive. Is there a risk that Rh disease can happen in this scenario? Explain. So remember, Rh disease happens if you have an Rh negative mother with an Rh positive baby. And it looks like this is the case. The mother is Rh negative and the baby is Rh positive. So there is a risk. Yes, there is. Now, the thing is, we don't know if this is a second pregnancy or first. Generally, if it's the first, what's going to happen is, as I mentioned before, the Rh antibody, the Rh protein will go into the mother's bloodstream and cause the mother to produce Rh antibodies. Now, it will not harm the first baby, but the second baby, because the Rh antibodies are there now, will get harmed. So there is a risk, especially if this is the second baby. If it's the first baby, it's just going to start to build up. Okay, so as I said there, we talked about genetics and that immune system interaction as well, which is very interesting. Ties a lot of concepts together. We're going to take a quick look at just how to donate and receive blood, because that's a common thing in society, and there's a great need for blood, and unfortunately those needs aren't always being met, and lives can't be saved all the time. So it's important to know at least about what what that means to donate and receive blood, and then how it works. So generally, you can be a donor, so that means you give blood, and you can also be a receiver. But you can't give blood just to anybody, and you can't receive blood just from anybody, or not always. So you'll notice that if you're blood type A, you're able to receive from blood type A or O. So any blood type A can receive from blood type A or O, regardless of that genotype, heterozygous or homozygous. Blood type B can receive from blood type B or O, regardless of whether it's homozygous or heterozygous. And blood type AB can receive from basically all of them, A, B, AB, and O. And then you'll notice, unfortunately, blood type O can only receive from O. So you can take a look at this, but there is definitely someone who's a universal donor, meaning someone who can give to everybody. And so if you look at this column here, you'll see O can basically be given to everybody. So O is usually a universal donor. And then universal receiver, that's basically someone who can receive from anyone. So you'll see A is pretty picky, B is pretty picky, O is pretty picky, but look, AB doesn't really care that much. So AB is generally a universal receiver. Now, we're going to go over that in some detail to see why is it the case. Why can O be given to everyone and AB be received, or AB receive from everyone? And why is it that O just doesn't like anybody else? Why can't O interact with other blood types? So basically, what happens is if you try to, if you inject blood into a person that you're not compatible with, that's going to cause issues like coagulating, your blood basically like clumping up, and that's not good. A technician who draws blood, by the way, there's another little career connection, is called a phlebotomist. So there's an art to that. There's knowing how to do it properly without bruising as well, just like with giving a vaccine. And it's a very important role because you need to test blood for a variety of different reasons. So let's go take a look at why is it that there's certain matches that you have for donating and receiving blood. And once again, it basically comes back to the immune response. So your immune system wants to protect you. If something enters your body that it's not used to, your immune system is basically going to try to destroy that thing. So your immune system makes these defense proteins called antibodies, okay? Kind of like we saw in the RH disease. When the mother that's Rh negative gets some Rh proteins in her bloodstream because of the baby, the mother's going to produce these Rh antibodies, which will destroy the Rh invader that it's not familiar with. Okay, so your immune system produces antibodies to protect itself from things that it's not used to, and those things it's not used to, they're called antigens. Okay, those are the foreign invaders. So suppose you're a person with blood type B. If you're a person with blood type B, let's go to blood type B over here. Your immune system recognizes these B carbohydrates, but it doesn't recognize any other carbohydrates. Okay, so let's say all of a sudden you get some blood type A, you see basically a triangle come into your body. And so your body is now going to produce this antibody called anti-A to destroy that triangle because it doesn't like it there. So here you can see a result. Basically, you have, if you're type B and you put type A in there, your blood is going to basically clump together because you've produced the antibody to destroy the type A carbohydrate, which is the antigen that came into your body. And same thing with receiving AB, right? If you receive AB, the B part's fine. Like your body will be like, okay, cool, there's some B, but wait a minute, hold on. I see you brought in some A as well, and that's going to clump up as well, okay? But if you receive B, you're fine because you recognize B. Interesting, if you receive O, you're also fine. What do you think that is? Look at O. Ah, yes. O doesn't really have any strangers on it. There's no carbohydrates on it, so it just kind of comes in sort of undetected, so you're able to receive it. Okay, and so if you look at A, it's kind of the same idea. A recognizes A, so it can get some A. O doesn't have anything on it, so O can come in. But as soon as you throw B or AB in there, we make these anti-B antibodies to destroy the B, the circles, because we don't recognize it, right? The B is an antigen in this case. So this is sort of the The antibody you create, the antibody recognizes the antigen over here, and then basically targets it for destruction. So let's say, for example, this is a type B. This is a B antibody. Okay, so this is a type A person. Let's say this is a type A person. They will make a B antibody when B blood enters them. And if this is the B blood, then the antibody will recognize the B blood antigen and basically target it for destruction, which is kind of interesting. So that's called the immune function, the immune system function. Really cool how you produce these proteins to basically protect you from danger. Even though we're trying to save you, like if your system's not used to it, we're going to destroy it just in case. If you look at type O, basically it just coagulates for every single blood type except for O, because O doesn't have any carbs. So everything is a stranger to it. The A carbohydrates, the B carbohydrates, the AB combination, all a stranger. But AB, if you look at it carefully, it knows triangle, it knows circle. So you can put in A, B, and it's okay. And O, because there's nothing there. So really the AB is not really going to produce any antibodies. Okay. And this also applies for the positive, the Rh positive, negative. So, for example, if you're blood type O minus, O minus can be given to everybody because there's no Rh factor at all, and there's no carbs. Okay. If you're O plus, you can be given to most people, but you can't be given to people who are minus, because the plus there will be recognized by the minus people and then destroyed because they don't have any plus protein in their body. So it's really that minus, O minus, that's a universal donor because, it's sort of the most inclusive because people who are getting O plus, if they see a plus and they don't have the plus in their body, that's basically a sign that there's a stranger coming in and they have to destroy it. So here is a summary of the universal donor and receivers. And so you'll notice that the blood types are listed here, the genotypes. You can see the antigen that they present, meaning like their carb. We call that the antigen because that's how your body would recognize it. So A has an A antigen, B has a B antigen, AB has both. O, just there's no carbs on it. What antibodies would... you can see the antibodies and who you can donate to and receive from. So if you're a type A person, you're going to make antibodies to B because you don't like B. If you're a type B person, you're going to make antibodies to A because you don't like A. If you're type AB, you're not going to make antibodies because you get along with both. And if you're type O, well, you don't get along with anybody, so you're making both antibodies. And so basically, who are you able to receive from? If you're type A, you can just receive from A and O because you made antibodies that'll destroy B. If you're type B, you'll receive from type B and O because you made antibodies that'll destroy A. If you're type AB, you'll receive from everybody because you don't make any antibodies at all. If you're type O, you'll only receive from O because you made antibodies to A and B. You don't get along with anybody. Who can you donate to if you're type A? Well, just for the similar reasons to A and AB because they recognize you. If you're type B, you can donate to B and AB because, again, they recognize you. If you're type AB, only to AB because, you have an enemy to A and an enemy to B. So you can only do AB. And if you're type O, you can donate to everyone because, again, you don't have any of the antigens, you don't have any of the carbs, so no one really recognizes you. You kind of sneak in. But really, it's primarily O minus, right? O minus is that universal donor. And the universal recipient technically is AB positive because if you're AB negative and you get some positive blood, that's not good. So AB positive is universal recipient. So again, a great connection with our immune system function.